It is a great honor and privilege for me to be invited to speak to you, to provide you with an update, a review of some immunological aspects of the interaction of PRRS virus with pigs, to give you a bit of perhaps new information about the immunology, and then build on the immunology to look at where we are in vaccines against the virus, and what makes a vaccine against PRRS that is likely to work, and things that we might do to make it even better than it currently is. The work that I will be presenting to you, of course, is not done by me. It's done by outstanding students and scientists in my laboratory and other laboratories around the world. I show some of those individuals here. I want to point out Li Juan from China Agriculture University. She was a former student of Yanhan Chen. Faculty and scientists from many universities, University of Illinois, South Dakota State. Not shown on here is Jeff Zimmerman, who we've worked with some wonderful studies in the past, and so on. PRRS is a very important problem everywhere, and there are a number of agencies and organizations in the United States that are eager to fund research on PRRS virus. And I want to disclose that I have 2 patents on the virus, and I hope that those do not affect anything I say today. Okay. We'll talk first about the pathogen, the virus that interacts with PRRS. This is an incredibly genetic diverse group of viruses. Type 1 European viruses have enormous genetic diversity, and they're very different from type 2 viruses. The virus we're primarily interested in Asia is the type 2 virus, because this is the virulent virus that really causes disease and economic loss. And the reason that the type 2 viruses are highly important is that they grow to higher levels than type 1. Peak viral loads are much higher in susceptible pigs. There's marked respiratory virulence. Respiratory virulence in type 1 viruses tends to be low. Very high reproductive virulence. Come on here now. Okay. Compared to type 1 viruses. And airborne transmission has been well documented in type 2 viruses. It was many years before airborne transmission was documented in type 2 viruses, and the reason was it was looked at very early in studies involving type 1 viruses, and no airborne transmission was noted under any condition at all. So it turned out to be a big surprise that this is a major route of transmission for type 2 viruses. The infection of PRRS virus in pigs is widely shown to look something like this, where following infection there's a rapid rise in virus in the blood. Viremia lasts at a high level for a few days, then begins to decline. The total period of viremia is quite long, 4 or 5 or 6 weeks. Eventually it goes negative in the blood, but it stays hidden in lymphoid tissues. And in this course of viremia, we are interested in what controls the primary infection here. In contrast to many viral infections, it does not appear to be neutralizing antibodies, because they tend to show up at time periods after the viremic infection has been resolved. This period of long viremia is noted in young growing pigs, where most of the studies are done. So here, virulent virus infection, rapid rise to high level of virus, prolonged viremia shown here for 28 to 35 days, disappears from the blood but is present in lymphoid tissues. Attenuated viruses, which are used for vaccines, show a less pronounced ability to grow in pigs because they're attenuated. They still have a long viremic period. if we compare these same two virulent and attenuated viruses in sows, we get a very different picture. Sows virulent virus grows much less well for a much shorter period of time, and attenuated viruses grow even less well for about the same period of time. This has big implications if in vaccinology you do not want to cut vaccine doses if your virus is attenuated for growth in sows. This difference in growth in young pigs versus old pigs is due to the fact that virus is more, that the macrophages of neonatal or young pigs are more permissive to virus growth, as shown here, compared to the macrophages taken from adult pigs. So there's this innate resistance to infection that is present in older pigs compared to younger pigs. Now, the prolonged period of viremia, the low level of neutralizing antibodies seen in young pigs, led people to think early on that the immune response to PRRS virus was not very good. Okay. And we and others have looked at the immune response. It's commonly assumed that cell-mediated immunity is an important mechanism for controlling viral infections, but we were unable to show any relationship between cell-mediated immunity and viral infection. Here we're looking at lymphoid tissues of pigs in acute infection or in persistent infection, where we documented the amount of virus in various lymphoid tissues and the amount of interferon gamma secreting cells, the best measure in pigs of cell-mediated immune responses. And clearly you can see that there is no relationship, either in acute infection or in persistent infection. And the interpretation here is kind of tricky, because one explanation is that T cells are not important in PRRS virus immunity. That would violate everything we know about immunology and resistance to infectious disease. Therefore, it's more likely that this assay that's being used is not able to measure the relevant T cell response to virus. But if we do not have good measures of T cell immunity, right now our ability to understand T cell based mechanisms simply has to wait until we get better at measuring T cell immunity in the pig. But our tools for looking at antibody responses are pretty good. And we see here that there is a rapid rise, typical of most antibody responses to antigenic substances. Here we're looking at non-structural protein 2 antibodies, nucleocapsid antibodies, envelope glycoprotein antibodies of different classes. And we see that here is our pattern of viral infection. By the peak of infection at 7 to 10 days, we are already starting to see an increase in antibody levels. So this is not an inability of pigs to mount an immune response to virus. And I highlight nucleocapsid here because this is the primary antigen in ELISA assays that are used to monitor herd health programs with respect to PRRS virus. And again, we see that nucleocapsid responses are detectable at about day 10 or so, go to high levels, and then decline over time. These antibody responses we measure in serum come from antibody being secreted by virus-specific plasma cells. Those plasma cells appear rapidly. They peak at about the time that viremia is being cleared. We see high levels in spleen, in sternal lymph nodes, sites where the virus is present in large amounts. And we see that they recognize all the different various proteins of the PRRS virus. So this is the immediate primary response to infection. The memory response, cells that are specific for different viral antigens that wait until they see antigen again, those are also being produced in very large amounts in all of the tissues of the pig where virus is present against all of the different proteins of the virus. Memory is believed to be the immunological basis for how vaccines work. So we see that memory cells are abundant, especially where virus is present. So we know that live virus induces a strong memory immune response, and this response is not impaired by viral infection. Okay. All of this suggests that, in fact, the immune response to PRRS virus is much better than people used to think. Here we were looking at antibody responses to individual viral proteins. The functional responses are important because the neutralizing response in particular is assumed to be the mechanism by which antibodies work. And it's been well documented that in growing pigs, neutralizing antibody levels tend to be low. Here we're looking at homologous and heterologous neutralizing activity where pigs were infected with different PRRS viruses. Serum was taken, and the ability of that serum to inhibit or neutralize the same list of viruses on the x-axis is shown here. Homologous protection is in the black bars. We see some combinations of virus and serum where we see low levels of neutralization, or some where there's little or none at all. So here, MN184, no neutralizing antibodies at all, even though in these infections viremia is cleared. And heterologous protection, very little evidence of heterologous neutralizing antibodies, even though we know that if pigs see PRRS virus, they're substantially resistant to reinfection with another virus. But it turns out that the neutralizing antibody story is more complicated than was appreciated when studies were only done on growing pigs. We have gone out to sow herds in Minnesota and taken virus samples from herds naturally infected with virulent PRRS viruses and noted that there are high levels of broadly neutralizing antibodies, heterologous neutralization. So here are the mean and standard error in neutralizing titers of 20 different sows in a herd, where we see that the average 50% neutralization titer is about 1 to 500. The previous slide, the highest titer that was shown in the entire figure was 1 to 16. In a second herd, we had a similar observation, 14 animals. All of them have neutralizing antibodies, broadly neutralizing antibodies, with a titer of about 1/80. And I want to emphasize this cross-neutralization activity shown in the bottom left, where in herd number 1, the viruses that were in that herd are circled in yellow, and they are about 15% different from the virus circled in orange, which was the target virus being neutralized by the serum. So again, this is cross-neutralizing activity at high levels. I just showed you neutralizing activity against VR2332. This neutralizing activity can be demonstrated with any type 2 virus we test. A number of those are shown here. What was even more surprising to us was that this activity is broadly neutralizing against type 1 viruses as well. So this tells us that while neutralizing antibodies are not required for control of PRRS virus, they may have a role to play. So it opens up a new opportunity in the immunology of PRRS virus to look at new ways in which we might be able to stimulate better protection. So it's clear we have neutralizing antibodies against virus. What is the target? Target has always been assumed to be GP5, the major envelope glycoprotein. And I could show you many papers that say that that is what the target is. When Joanne Lee looked at this in our lab, our approach was to make the recombinant proteins of PRRS virus that have GP5 in them, the heterodimer GP5M, the ectodomain portions of GP5 that are outside the virus, or the membrane protein by itself, the portion outside the virus. We put these proteins on a column and use those to specifically purify only the antibodies that bound to these forms. So here we ran serum over the column, we washed it, we specifically eluted the antibodies. We have specific antibodies here. Anti-M antibodies only bind M. Anti-GP5 antibodies only bind GP5. And antibodies purified against GP5 and M specifically recognize both proteins. So, do these purified antibodies specifically selected to these outer membrane proteins neutralize viral activity? Joanne showed here that these antibodies bound virus. She mixed the antibodies with on beads to the virus. If the virus bound to the antibody, it would precipitate with the beads. And so here we're looking at RNA copies in pellets, and we see that GP5M binds. We see that GP5 by itself binds virus. M does not. Non-structural protein 2 does not. Okay? But, so then, does this binding block infectivity? And the answer at the bottom is no. Here we're looking at nucleocapsid in infected cells. As we add increasing amounts of antibody, we see no decrease in the amount of nucleocapsid, no change in the infection level of the cell. So we're starting to make progress now. It's quite clear to us that the target of neutralizing antibodies is some other protein, which we're very interested in investigating. So we have the immunology in PRRS now is it's clear that there is an immune response. It's pretty good if you look and measure what is happening, but we do not know what the mechanism of effective control is. We do not know what the targets of antibodies are on the virus. We do not know the role of cell-mediated immunity, but we do know for sure that the immune response to PRRS virus in pigs is completely effective. And that has been shown time and again now by this method called herd closure. Herd closure, or load, close, homogenize, a practice that North American veterinarians invented that completely gets rid of PRRS in pigs. So if you fill a barn up with pigs and you homogenize them with vaccine, you make sure every single pig is infected, so there are no reservoirs of future infection, and you wait. You wait 250 days or so. You bring new pigs in, they do not get infected. It's clear that if you wait long enough, pigs completely eliminate viral infection. And that's really important because it says that the immune response to PRRS virus works. It says that our goal will be to find conditions where vaccines work faster. Rather than taking 250 days, can we induce immunity in pigs so it takes 20 days or 50 days, that it works better? So I would like to turn now from immunology to vaccinology and look at let's talk about vaccines a little bit. And the first thing, just to remind ourselves about, is that there are lots and lots of vaccines against PRRS. This is a list that Marika Gensau and Berenger and I made a few years ago that showed vaccines around the world before this huge vaccine production effort ensued in China. So the list is probably twice as long now. So there are lots and lots of PRRS vaccines. But most of these are no good. They do not work, okay? And I'd like to go now and talk a little bit about what qualities are that good vaccines have. So when we, in the future, when we talk about vaccinating for PRRS and the virtues of vaccination and what to expect and try to understand what goes wrong, we're only going to talk about good vaccines, okay? One of the qualities, and so we're gonna, these are the conclusions now, and we will look at data, but it's very clear that good vaccines that work against PRRS have to be able to grow attenuated live virus. Very clear that killed products and subunit products and vectored products and third-generation products do not protect pigs against PRRS. These viruses have to be, good vaccines have to be safe. It's no good to vaccinate animals if the vaccine itself is going to cause disease. Attenuated viruses have been modified to be safe. Virulent field viruses, under some conditions, can induce good immunity, but you're maintaining a pathogenic virus in the herd. And to show safety, it's very important that the model be the late gestation sow. This is the most sensitive animal to bad effects of viral infection. Okay. So, if we look to the right, the modified live viruses have to be cross-protective. Cross-protective immunity is fundamentally important. It's obvious. Any vaccine now today consists of a single virus in a bottle. That single virus has to be able to protect against all the viruses in the field. So if you're going to look at and claim vaccine efficacy, it's really important to show the efficacy with field viruses, not against the parent strain of the vaccine. Okay. And lastly, good technical support. The PRRS virus is a difficult challenge in maintaining good swine health. Knowing how to use the virus, when to apply it, what the important conditions are, really matters in being able to get products that have efficacy to get them to work well. So having good technical support is important. Okay. We're back. Okay. So, this is a study looking at efficacy in killed vaccines, and we do not need to go through the data. We simply have to read the title of the paper. Failure of an inactivated vaccine to protect gilts against heterologous challenge. They used the right model, heterologous challenge. And it was gilt, so they're looking at the reproductive model. And it did not work. Okay? Killed vaccines do not protect against PRRS. Here's a study looking at a subunit vaccine. The subunit was GP5, major envelope glycoprotein. Again, You do not need to look at the data, just read the title. Immunization with a major envelope protein, enhanced clinical disease following challenge. This is worse than no good at all. It made things worse. Subunit vaccines do not protect. Not only did they not protect, they exacerbated disease. Okay. And this is a study done in the US in a pregnant sow model, late gestation, where if we look on the right side, there were 5 groups. We're comparing an attenuated modified live virus to a vectored product, which was GP5 and M, and a subunit product, which was virus-infected cell lysate with extra GP5. And there were 3 endpoints here shown here. Were the sows PCR positive? Were they viremic at 8 days? And we see animals infected only, shown in red, with no vaccine product. They were viremic. No significant difference if they had vectored vaccine or subunit vaccine. They were all protected from viremia at 8 days by modified live virus. Were sows feeling bad enough that they did not eat? Interestingly, the virulent virus challenge, the sows kept eating, but if they got the vectored or the the subunit vaccine or the vectored vaccine, it made them sicker. It enhanced disease so that they stopped eating, completely protected from going off feed by modified live vaccine. And key production parameter, pigs failing to wean, pigs that could not survive to weaning. 58% of the pigs challenge only failed to wean. Numbers were increased by, again, the subunit and the vectored vaccine. A significant reduction in if they got live virus. And let's note here that this was not 100% protection. It was a quantitative reduction. We will come back and look maybe at some of the reasons why that might be, because it's an important issue in understanding how PRRS vaccines work. What about the previous study was cross protection, but we didn't show any viral information. This is a study done by Shishan Yuan before he was working at Boehringer Ingelheim, looking at cross protection against high-path PRRS virus. We all know this is a very virulent virus. He chose 3 groups here. Challenge only, 8 of 12 pigs were dead at day 21, and whereas vaccinated with a modified live virus, no death. And we see at the bottom left that pigs started dying at about day 6 or 7 in the absence of vaccine, and by the end of the experiment, again, 2/3 of the pigs had died. We also saw substantial protection from clinical signs, shown on the right-hand side, fever and other scores of illness, substantial reduction in vaccinated pigs compared to non-vaccinated. Again, these are not 100% differences, but it's clear that there is obvious protection, not 100%, but obvious protection if pigs have seen a virus once and then are challenged later on. Vaccines provide protection. And this cross protection has been seen in every challenge study that I have ever had the opportunity to look at directly or in published papers. These are a series of studies done at Boehringer Ingelheim involving a series of virulent field isolates that are substantially genetically different from either of the 2 attenuated viruses used in a vaccination trial. To the right we see that in the absence of vaccination, percent lung lesion scores are substantial in every case. Note that in experiment to experiment, there's variation, but there's always disease. And these are the averages of 10 animals per group. And we see that if they were vaccinated first, we see, again, there's variation, but in every case, you see less disease. And the percent reduction is shown here. And just to confirm that there is no relationship between the genetic similarity of the vaccine to the challenge virus, the data were plotted here where percent genetic difference is on the x-axis and reduction in lung lesions is on the y-axis. And we see, note again, a lot of variation if you run this trial many times, whether the viruses are 10% different or 13% difference. But on average, there's no relationship between the genetic similarity of the vaccine virus and the virulent virus. These experimental studies of cross-protection can be reproduced in the field in very large trials. Here we're looking at an example of using vaccine in an endemic infection system to see if you can treat PRRS virus and reduce or control disease losses due to PRRS. So in this system, over a 2-year period from 2003 to 2005, in this all-in, all-out system, nurseries were filled with pigs. They stayed there for a period of time, and then they all were taken out. And what is shown in the data are how many fewer pigs went out than came in. So this is mortality. And we see mortality between about 5 and 15% consistently over a 2-year period. They finally got tired of this. They instituted a vaccination program at this point, and over the next 2-year period until 2007, there was a very consistent and reproducible and sustained reduction in mortality by 50%. And this level of mortality was the same as in other nurseries in the same system that had no PRRS at all. That is shown on the bottom. Okay? And we're looking at over 300,000 pigs in these two cases. Lots of data. Mortality, no difference. Average daily gain, no difference. Feed efficiency, no difference. So here in a commercial field, realistic setting of virulent PRRS virus in these nurseries, we see that vaccine worked quite well. But what else do you notice about this example? This addresses two other issues that come up from time to time in vaccines for PRRS. One issue is reversion to virulence. You cannot imagine how many times I have been asked in settings like this about reversion to virulence, and to the point where I'm tired of hearing the question, because it does not happen. Here is a case. Vaccination for a 2-year period, hundreds of thousands of piglets, no reversion to virulence. You do not see an increase in disease occurring here due to reversion to virulence. It simply does not happen. You can find examples where vaccine viruses recombine with field viruses. You can find examples where vaccine viruses are maintained in pig farms because of the way the pigs are being managed, but you do not see cases where vaccine application to control or prevent PRRS causes disease because the vaccine reverts to virulence. I'm not saying it can never happen in the history of the universe, but it is if it does, it is such a rare event that the benefit of vaccination is so much more important that it would not matter if it happened. Second thing you see here is you do not, so here we have an endemic situation. These pigs have seen virus, they have antibodies, vaccine is put into the herd. You do not see an increase in disease caused by antibody-dependent enhancement. The concept of ADE, antibody-dependent enhancement, is that low levels of neutralizing antibody will incompletely bind to the virus, and because the virus infects macrophages, this antibody-virus combination binding to the antibody receptor on macrophages will increase infection. It explains certain features of dengue virus infection in humans, but it does not happen in the field. I am aware that there are publications about ADE in the literature, but it does not happen in the field, and it's not up to me to explain why people think they observe it in the laboratory. Okay, now I want to come back to this issue of variation we've seen, because there are limitations to PRRS vaccines. They do not replace sanitation, good management. They do not replace good genetics in your herd. They do not replace good animal care. And they do not overcome variation in the ability of pigs to respond to PRRS virus. And that variation is shown, in my opinion, quite clearly in this example, where groups of 10 pigs were infected with individual different viruses that range from highly virulent to very low virulence. But if you looked at the lung lesion scores in individual pigs, in every group, even low virulent, you found variation. 2 pigs here showed high levels. In group 4, high disease, 1 pig died. You had another pig completely resistant, no lung lesions at all, and others with very low levels. So this variation in the ability of pigs to respond to viruses, to vaccine viruses, is something that we need to understand in order to get more uniform efficacy of vaccines. The second major limitation you need to understand about PRRS virus vaccines is they only work against PRRS virus. Okay? So, when you have abortions, is that caused by PRRS virus? If you have respiratory disease in the finisher, is that caused by PRRS virus? Because you know that there are other things that cause abortions, that cause respiratory disease and pneumonia. So you have to have good diagnostics to confirm that the problem is PRRS. Because if it's not PRRS, a PRRS vaccine will not help. And I want to say a word or two about vaccines, because many people's ideas of looking at virus is to ask, is it present or absent? Or looking at immunity at the IDEXX ELISA and asking, is it positive or negative? You would never ever go to your financial person or look at your balance sheets and ask if you were making money or losing money. You will ask, how much? How much are you making? Okay? You will never talk to your nutritionist and go to the feed company and ask, are my pigs growing or shrinking? You want to know how well they're growing. It's quantitative. What's the average daily gain? What's the feed efficiency? And reproduction is the same. Did my sows give me piglets? You don't care. You want to know how many. How many pigs per sow per year? How many pigs wean? The diagnostics need to be quantitative as well. You have a PRRS virus infection. How much virus is in the sample? This gives you information about the timing of infection. You can get PCR positives a week before their seroconversion. So if you're screening by PCR and you get low levels of virus in a herd that's serologically negative, that's a clue that virus just got in, and you saw it right at the beginning. Vaccine, we've seen that vaccine viruses grow to much lower levels in pigs than virulent viruses. If you have a stable situation and you get low PCRs, is that because you vaccinated the herd and you're simply measuring the response to vaccine compared to the levels of virus in a virulent infection? In the same way for the IDEX ELISA, this is a sample to positive ratio where IDEX tells you that a positive is 0.4 or bigger. It does not mean that a 0.39 is negative. What is the background of the IDEX ELISA in negative herds that have never seen PRRS virus? Who knows? 0.3, 0.35, 0.2, 0.1? It's less than 0.1. If you're running IDEX ELISAs in your herd and it's negative, those numbers are going to be 0.1 and less. So if you're running your serology and you see a 0.2 or a couple pigs with a 0.2, you should be nervous, because there's a good chance those pigs are just starting to seroconvert, and they may continue to go up. So the numbers really matter, and your ability to understand and know what they mean improves the more you look at the numbers and compare those to what is going on in your herd. Okay. So the concluding statements I'd like to leave you with regard to the immunology and vaccinology of PRRS is, first of all, that the immune response to PRRS virus really is good. It is robust, it's strong, it's cross-protective. We don't know everything about this immune response, as you've seen, but for sure, it's pretty good, which suggests that PRRS virus is not suppressing the ability of pigs to respond immunologically, at least to their own infection. The mechanism of protection and the role of antibodies in viral clearance and memory is something that we are slowly figuring out. Okay? Why are all pigs not protected? Again, we do not have all the answers here, but it's quite clear that part of the answer is that there's a lot of variation in the ability of pigs to respond. And we do not know the key protective mechanisms. If we figure those out, then we can use those as correlates to better predict how pigs would respond to infection without having to go the trouble of actually giving them an infection. Live attenuated virus vaccine gives good cross-protective immunity. Okay? And this includes against highly pathogenic viruses such as HP-PRRS. Subunit and vectored vaccines do not provide consistent protection. I did not address the issue of, well, can they boost an immune response that you get with live virus? There's actually two sides to that question. One is that when you pay attention to what you're doing, you find out that live virus by itself works pretty good. There's not much room for improvement to try and make it work better. So experimentally, it's a real challenge. And the second is that some people have worked very hard to try and figure out if you could use an immune and boost model to get good protection, and no one has been able to consistently show that that works. So that it appears quite clear now that to get good immune responses to PRRS virus, you have to give them a live virus that can grow in the pig. And lastly, again, vaccines are more effective with good diagnostics. And you will be hearing more about diagnostics later today. You'll hear about a revolution in diagnostics that Jeff Zimmerman came up with that really gives us great opportunities to improve our ability to have a good understanding of herd health, not just for PRRS, but for all of the diseases of pigs. So again, it was a great pleasure to be able to share some of the information I have with you, and I look forward to the rest of the presentations today. Thank you.